All right, ladies and gentlemen, welcome back. So, last time we were talking about how we believe the first prokaryotes kind of came about and really how they ended up shaping the Earth as a whole. So, the next logical progression in life on Earth makes sense to start talking about eukaryotic cells. So, remember the big difference here. Prokaryotic cells are your cells that don't have any internal organization. They don't have a nucleus, they don't have organelles. Your eukaryotes, on the other hand, do have those structures. So, our oldest known fossils of eukaryotic cells date back to about 2.1 billion years. So, the question then becomes, how do we get from a living cell that has no internal structure to a living cell that does? And that's where we enter talking about this idea of the endosymbiont theory. And what this theory proposes is that mitochondria and plastids, so chloroplasts and that sort of thing, among other internal organelles, may have formerly been small prokaryotes living by themselves that ultimately got taken in by larger host cells. So let's sort of take a look at what this theory is all about. So let's break this down for a second. So when we talk about an endosymbiont, right, let's break that word apart. Endo being a prefix meaning within, and a symbiont or symbiosis, living relationships between two living organisms, right? So the thought is that the ancestors of mitochondria would have been some early aerobic heterotrophic prokaryotes, right? So they would have required oxygen, but they would have required obtaining materials from their environment in order to make their energy. Ancestors of things like chloroplasts would have been photosynthetic prokaryotes, so they would have been capable of carrying out photosynthesis and making their own energy by that means. So the theory suggests that you had a larger prokaryote and then these smaller prokaryotes living in the environment, and essentially endocytosis would have occurred. So endocytosis, right? A cell takes its cell membrane, envelops its food particle, brings that food particle in, and typically would digest that food particle and use it for energy or other source materials to build things that it needed. What seems to have perhaps happened in this case is that these ancestral mitochondria, these ancestral chloroplasts, or these free-living prokaryotes, got engulfed by a larger prokaryotic cell, but for one reason or another, that larger cell was unable to digest the prey that it took in. So instead, because these ancestral mitochondria and ancestral chloroplasts were capable of doing processes that that larger host cell was unable to do for itself, somewhere along the way, a symbiotic, more specifically, a mutualistic relationship evolved. And eventually, this happened enough times, and that symbiotic mutualistic relationship became so integrated within this bigger host cell that not only did those ancestral mitochondria, those ancestral chloroplasts, start depending on their hosts for materials, but that host cell also depended back on them for the ability to now generate their own food in the case of chloroplasts, or to probably more effectively produce energy in the case of those ancestral mitochondria. That this eventually became a strong enough dependence on each other that we now are talking what we know as eukaryotic cells today, and we can't separate out those mitochondria and chloroplasts and allow them to live independently outside that cell. And the reverse would also be true, that that main cell depends on those as well. And so we now talk about them as a single organism. And more than likely, this is what we assume would be serial endosymbiosis, meaning that this would have happened multiple times over and over and over again. And not only that, it would have created other folds and other structures within the cell as well. So the infolding of membranes to eventually develop the nucleus, to develop Golgi bodies, to develop your rough and smooth ER, so on and so forth. So sounds like a pretty logical, pretty elegant theory, but where's the evidence, right? Because anything that we're going to discuss as a scientific theory, that means there needs to be evidence to back this up. So one of the things that we point to as evidence for this particular idea is that the inner membranes of the mitochondria and the chloroplasts have enzymes and transport systems that are homologous to what we would find in living prokaryotes today, specifically in their plasma membrane, right? So that plasma membrane, remember, is their barrier from internal and external environments. So it's their outer covering outside of their cell wall. And so seeing enzymes and transport systems that are much more prokaryote-like as opposed to what we see in typical eukaryotic systems gives us some evidence to suggest that this is in fact how this happened. Cool thing about mitochondria and chloroplasts is that they are also capable of replicating within the cell, outside of the cell, going through cell division. And in fact, they do it in a much similar process to binary fission, which is how prokaryotes themselves replicate. Other cool thing, mitochondria and chloroplasts also have their own circular DNA molecule, again, structured much like the main chromosome in a prokaryote. So they actually have their own set of genetic material, which again would make sense if they were free-living organisms at one point. They would have needed their own genetic information in order to accurately replicate, as well as to carry out other life processes that were necessary. So where did you get all of your mitochondrial DNA in all of your cells? Cool thing is that it is all maternally inherited. So all of the DNA in your mitochondria specifically came from your mother, which is an interesting piece of information when we consider that your nuclear DNA is a 50-50 split from mom and dad, which means we'd have to start teasing out information from mom and dad to determine which genes came from which parent. Your mitochondrial DNA all came from your mother, and so we know that it's maternally inherited, and so it's another interesting thing that we can utilize when we start talking about analyzing DNA as a means of understanding heredity, ancestry, and so forth. But that's not where the evidence stops. They also contain their own tRNAs, ribosomes, both molecules that would have been necessary for protein synthesis within a free-living organism. These two particular organelles also carry out that. And those ribosomes are actually more similar in their structure to the ribosomes we see in prokaryotes. Once again, the eukaryotic ribosomes are slightly different structures because they've evolved and changed over the course of evolutionary history. Were we able to figure out who these guys were probably related to? Well, it comes back to RNA sequencing. So we believe that in the process of using modern organisms that exist, studying their RNA molecules, as well as studying the DNA that is present and the tRNAs and things like that that are present in these current mitochondria and chloroplasts in living eukaryotes, it is believed that alpha proteobacteria are the closest living relatives to our mitochondria today, and cyanobacteria are the closest living relatives to the chloroplasts. So, realistically, eukaryotic cells are just these genetic and structural, what they call chimeras. So, if you know anything about mythology, chimeras were sort of these organisms that were pieced together So things like head of a lion, body of a horse, that kind of thing. So they're pieced together from other organisms. Your nucleus probably evolved from an endosymbiont belonging to our domain Archaea. If we look at the three domains that make up the living world, we talk about bacteria, archaea, and then eukarya. So, interesting thing when we get to classification is we'll talk about the fact that archaea and eukarya domains are actually more closely related to each other than those archaea, which are prokaryotic, are related to bacteria. And that's sort of where the three domain system needed to evolve from sort of the older five kingdom method of classification. So your genome is probably a product of what we talk about as genetic annealing, which is the production of a new genome through the transfer of parts of genomes from one organism to another. Basically, we're this big hodgepodge of genetic information that would have gotten taken from all sorts of sources until it was combined into the genome in these eukaryotic cells. Basically, you are what you eat. So eukaryotes are consuming all sorts of various bacteria and archaea throughout their evolutionary history, incorporating pieces that ended up from the engulfed prokaryotes that they were eating until we get to the structure and genetic information that we see in eukaryotes today. And again, when we look at internal structure, the origins of others are still of active research, but probably something along those lines of you are what you eat. Things like the Golgi and the ER probably originated as infoldings of the plasma membrane. Proteins that we see in things like our cytoskeletal structure in eukaryotic cells are homologous to proteins that we see in bacteria. Things like the eukaryotic flagella and cilia may also have evolved from symbiotic bacteria somewhere along the way. So, kind of interesting things that are still being looked at, studied, and we'll see what sort of information comes of it. So, we get to these eukaryotic cells, and then eventually we start talking multicellularity. So while the first eukaryotic cell itself, free-living eukaryotic cell, seems to have come from fossils as old as 2.1 billion years old, we don't get the startings of common ancestors that are connected to multicellular eukaryotes until about 1.5 billion years. The oldest actual known fossils of multicellular eukaryotes show up about 1.2 billion years. And our larger organisms that we're typically thinking of when we think of multicellular eukaryotes don't appear in the fossil record until late Proterozoic